Boa noite. Em nome, então, da Academia Nacional de Medicina, eu queria dar as boas-vindas a todos à sessão ordinária da Academia Nacional de Medicina, de número 32, a 32ª sessão acadêmica deste ano, o ano 191º, e em continuidade ao excelente programa desenvolvido hoje à tarde, sob a coordenação dos acadêmicos Marcelo, Barcinski e Marcelo Morales. Nós vamos, então, agora dar início à sessão. E eu passo a palavra ao secretário-geral Ricardo Cruz, mas antes gostaria de submeter a ata que foi encaminhada a todos por e-mail e por correio, por WhatsApp. Se alguém tiver alguma correção ou comentário, por favor, a palavra está aberta. Não havendo, a ata está aprovada e agora o secretário-geral da Academia Nacional de Medicina, acadêmico Ricardo Cruz, vai fazer as considerações regimentais.
Excelentíssimo presidente da Academia Nacional de Medicina, acadêmico Rubens Belfort Júnior, excelentíssimo presidente por 3 gestões da Academia Nacional de Medicina, Pietro Novellino, queridas confreiras, queridos confrades, ilustres convidados, a Secretaria-Geral gostaria de assinalar que o Dia do Médico foi comemorado no domingo passado, dia 18 de outubro, e nós gostaríamos mais uma vez de ressaltar a homenagem feita à memória do acadêmico Idoberto Carneiro de Oliveira, um dos 265 médicos que perderam a vida durante a pandemia do Covid-19 e cuja história foi matéria no jornal RJ-TV da TV Globo. A Secretaria-Geral gostaria de, mais uma vez, externar aos familiares do acadêmico Idoberto Carneiro de Oliveira os nossos mais sentidos pêsames. Nesta segunda-feira, dia 19, em evento promovido pelo jornal O Globo, em homenagem ao Dia do Médico, a Secretaria-Geral gostaria de destacar a excelente participação da acadêmica Patrícia Rocco no debate sobre a saúde mental do médico durante a pandemia do Covid-19. Gostaríamos de parabenizar mais uma vez a acadêmica Patrícia Rocco pela sua participação, representando de maneira tão nobre a Academia Nacional de Medicina nesse evento. Já no dia 20, senhor presidente, terça-feira próxima passada, a sessão da saudade em memória do acadêmico Irã Silveira Lucas foi realmente emocionante. Eu gostaria de registrar que esta sessão contou com a prestigiosa presença da presidente da Academia Nacional de Medicina de Portugal, a acadêmica Fátima Carneiro. Da mesma forma, no próximo dia 10 de novembro, também uma terça-feira, também no horário de 18 às 20 horas, a Secretaria-Geral está preparando com todo o esmero a sessão da saudade em memória do acadêmico emérito Eustáquio Portela, e os oradores do primeiro bloco desta sessão serão o presidente acadêmico Jorge Alberto Costa e Silva, o acadêmico Antônio Egídio Nardi, que, como todos sabem, foi eleito na vaga do acadêmico Eustáquio Portela quando da sua passagem a membro emérito da nossa academia, e o terceiro orador será o acadêmico Adolfo Rores. A Secretaria-Geral gostaria de registrar a passagem no dia de ontem, com muita alegria, ontem, dia 21, do aniversário do nosso querido honorário nacional, José Marcos Fies. Amanhã, dia 23 de outubro, dia muito importante. Toma posse às 20 horas a nova diretoria da Sociedade de Medicina e Cirurgia do Rio de Janeiro, tendo como presidente o acadêmico Celso Ferreira Ramos Filho, e tendo na sua diretoria os acadêmicos Carlos Alberto Basílio de Oliveira, José Galvão Alves, José Suassuna e Paulo Niemeyer Filho, a nova diretoria da Sociedade de Medicina e Cirurgia do Rio de Janeiro. Dia 27 de outubro, às 10 horas da manhã, será a vez do acadêmico Maurício Magalhães representar a Academia Nacional de Medicina em evento também organizado pelo jornal O Globo, o evento tradicional, os encontros, o Globo Saúde e Bem-Estar, numa comemoração do Outubro Rosa. E, finalmente, presidente Rubens Belfort, a Secretaria-Geral gostaria de registrar que na próxima quinta-feira, dia 29 de outubro, a 33ª sessão deste complicado 191º ano acadêmico da Academia Nacional de Medicina, onde nós faremos uma sessão espetacular, coordenada pelo presidente da secção de cirurgia, José de Jesus Camargo, com o presidente acadêmico Pietro Novellino e o secretário da secção de cirurgia, o acadêmico Rossano Fiorelli, numa sessão intitulada A Cirurgia Brasileira no Mundo. Nós teremos nessa sessão, a sessão Recentes Progressos, que contará com a participação do acadêmico Carlos Eduardo Brandão, e haverá ainda o lançamento do livro do acadêmico Rossano Fiorelli. Essas foram as comunicações da Secretaria-Geral, senhor presidente, nesse dia 22 de outubro de 2020, 32ª sessão ordinária da Academia Nacional de Medicina.
Muito obrigado, acadêmico Ricardo Cruz. Antes de abrir a palavra aos acadêmicos, eu queria comunicar que também no próximo sábado nós teremos uma sessão da Associação Latino-Americana das Academias de Medicina, onde o Brasil será formalmente eleito para a presidência da associação para o próximo biênio. Esta associação reúne as academias nacionais de medicina de países da América Latina, de Portugal e da Espanha. Será uma outra oportunidade para nós nos relacionarmos com os países todos dessa associação e tratarmos de coordenar nossas atividades e mostrar o interesse e a importância que o Brasil dá para esse tipo de trabalho, realçando as evidências científicas na medicina. E tratando de mostrar o valor das evidências científicas da medicina, eu quero fazer eco ao elogio que foi realizado pelo acadêmico Ricardo Cruz em relação ao trabalho da nossa acadêmica Patrícia Rô. Foi já comentado na parte inicial desse programa científico o sucesso do trabalho desenvolvido pela acadêmica Patrícia Rocco, tratando de coordenar um estudo rápido, eficiente, sobre a ação da nitazoxanida para reduzir a carga viral na fase precoce da Covid-19. Esse trabalho, segundo a professora Patrícia Rocco nos comunicou, já foi enviado para publicação e será logo disponível aos acadêmicos e à comunidade científica para conhecimento dos detalhes. Então, acadêmica Patrícia Rocco, nossos parabéns. É um exemplo que precisa ser divulgado para ser conhecido e copiado por todos. Eu passo a palavra agora à comunicação dos acadêmicos, aqueles interessados em fazer uso da palavra, por favor, levantem a sua mão biológica ou eletrônica.
Presidente.
Presidente Pietro Novellino.
Excelentíssimo senhor presidente Rubens Belfort Júnior. Senhor secretário Ricardo Cruz. Senhoras acadêmicas, senhores acadêmicos, organizadores desse belo simpósio, Marcelo Morales e Marcelo Barcinski, cuja atuação da acadêmica Patrícia Rocco foi enfatizada pelo nosso presidente e pelo secretário-geral. Senhor presidente, eu vou pedir vênia a Vossa Excelência para fazer um pequeno comentário sobre o Dia do Médico, que evidentemente foi domingo e a academia comemorou na quinta-feira que antecedia essa data. Eu, sem querer usar um ditado corriqueiro, não estou aqui para rasgar sedas, mas eu queria enfatizar o pronunciamento do acadêmico Ricardo Cruz, viu, que representou Vossa Excelência e nós todos com um pronunciamento curto, objetivo e por isso a Academia comemorou o nosso Dia do Médico. Eu também queria complementar uma pequena observação que eu fiz sobre Lucas, sobre São Lucas para alguns, ou Lucas, nosso patrono, quando ainda criança, ao ser indagado pelo nobre Diodoro, se teria, por acaso, algum projeto quando fosse adulto, respondeu: encontrar o Deus desconhecido, e a melhor maneira de servi-lo será a do médico. Então, senhor presidente, muito obrigado, viu? Embora já tenha passado, mas não poderia deixar, com a sua aquiescência, de fazer esse breve comentário. Muito obrigado.
Professor Pietro Novellino, sempre oportuno e atual. Muito obrigado. Não sei se existe mais algum acadêmico para fazer algum comentário. Antes do acadêmico Celso Ferreira Ramos se manifestar, eu já tinha programado fazer um comentário para elogiar a posse do nosso acadêmico amanhã. A Academia Nacional de Medicina se sente muito honrada de ter sido convidada para estar presente, e lá estaremos. É muito bom vê-lo aqui, acadêmico Celso Ramos, recuperado e forte. Vamos lá.
Muito obrigado. Eu queria começar exatamente. Eu tive que fazer uma inspeção local na unidade coronariana da Casa de Saúde de São José e quero agradecer o auxílio luxuoso do professor Cláudio Benchimol, que conseguiu fazer com que eu saísse de lá em coisa de 24 horas. Foi uma coisa rápida, nada que uma amiodarona na veia não resolvesse. Tudo bem, tudo tranquilo. Mas agradeço a sua manifestação também. Eu queria convidar todos eu creio, receberam por WhatsApp um convite para a cerimônia de amanhã. É um momento, para mim, de uma gravidade extraordinária, porque não quero adiantar o discurso, que, aliás, eu vou ter que sair em breve, porque eu ainda não terminei. Não quero adiantar o discurso, mas nós estamos enfrentando tempos muito complicados e complexos, dos quais essa academia tem sido testemunha, por vezes, involuntária. Mas o fato é que estamos numa situação muito ruim, estamos numa situação muito complicada, e assumir a responsabilidade da direção de uma associação que tem quase 200 anos de idade, quase 200 é um pouco de exagero, tem quase 150 anos de idade, foi fundada em 15 de fevereiro de 1886, é algo realmente de uma responsabilidade muito grande. Não quero adiantar, quero só vir aqui basicamente para pedir a todos que, na medida do possível, comparecessem, e eu espero, o presidente Belfort já nos garantiu o apoio pessoal e institucional dele e da academia, e eu gostaria de solicitar o apoio de todos os colegas e confrades, confreiras, nessa empreitada em que eu estou me metendo, nessa camisa de 11 varas que eu voluntariamente estou vestindo. Muito obrigado a todos.
Muito obrigado. Antes de passar a palavra aos acadêmicos Marcelo Barcinski, Marcelo Morales e Patrícia Rocco, para fazer a devida apresentação dos nossos próximos convidados, eu queria repetir aqui uma informação que eu recebi hoje de um colega cirurgião de Recife, Cláudio Lacerda. Por coincidência, ele ganhou um dos prêmios da Academia esse ano. Ele publicou no Jornal de Recife um artigo curto, onde basicamente ele conta que um jovem assistente da equipe dele estava de plantão quando chegou uma menina com quadro grave após haver sido transplantada do fígado e em parada cardíaca, foi atendida por esse jovem que, não tendo nenhum outro recurso terapêutico, mesmo sabendo que a menina era COVID positiva, fez respiração boca a boca na menina. E manteve a respiração boca a boca até chegar ao devido equipamento e a menina foi salva. Sem entrar no mérito da indicação correta ou não, esse é o espírito da medicina. O jovem pulou em cima da menina e tratou de salvá-la, colocando a vida dela acima de qualquer coisa. Isso é uma atitude tão brilhante, tão fantástica, que eu, com a aprovação da comissão que cuida dos jovens talentos em medicina, gostaria de especificamente convidar esse médico para fazer parte do programa. Esse tipo de atitude, onde o indivíduo coloca a vida dele em segundo lugar, frente a um paciente, não pode passar em branco. Então, eu achei que era minha obrigação divulgar isso aqui. Vou pedir que o acadêmico Ricardo Cruz coloque também no Confrades, porque não importa entrar no mérito se isso podia ou não, devia ou não ser feito, mas é a atitude médica que eu acho que precisa ser sempre valorizada. Então, eu acho que não há, espero que não haja comentário contrário, e não havendo, eu vou passar a palavra, então, ao acadêmico presidente da seção de Ciências Aplicadas, Marcelo Barcinski, para continuar esse programa científico me representando. Marcelo.
Muito obrigado, excelentíssimo senhor. Estão me ouvindo?
Perfeitamente.
Senhor presidente da Academia Nacional de Medicina, Rubens Belfort Júnior, é um prazer, então, dar continuidade a esse simpósio, que, como eu já frisei na parte anterior, foi uma ideia, uma concepção e uma organização do confrade Marcello Morales e que correu, enfim, de uma forma extremamente bem-sucedida, extremamente dinâmica, extremamente produtiva e continua agora, então, nessa segunda parte. I'll now give my thanks for Dr. Levine. Are you hearing me?
Yes, I hear you just fine.
Yeah.
Are you hearing me?
Yeah, I'm hearing you. I want to thank you for accepting our invitation and to being here lecturing for us. But I'll ask Dr. Marcello, who is the organizer of the meeting, to introduce you more properly. Please, Marcello. Marcello Morales.
Obrigado, Marcelo Barcinski, confrade Marcelo Barcinski. Agradeço a oportunidade, senhor presidente Rubens Belfort, por poder organizar essa sessão e compartilhar com os senhores as perspectivas das terapias avançadas. Senhor secretário Ricardo Cruz, muito obrigado também. Antes de fazer a apresentação do Dr. Levine, eu queria só salientar a angústia que todos nós temos nesse momento tão grave da nossa história e todo o esforço de todos os médicos e pesquisadores e que estão colocando todos os seus esforços para salvar vidas. Então, esse simpósio, essa academia, coloca à frente, como o nosso presidente acabou de falar, os médicos e pesquisadores colocam as suas vidas à frente para colocar o melhor do seu conhecimento para salvar vidas. E isso é um ímpeto dos médicos e também dos cientistas. Então, parabéns a todos. Aqui eu agradeço à professora Patrícia Rocco pelo seu empenho e pela sua coragem de ter colocado esse desafio à frente e ter levado, e isso foi um trabalho de cadastramento das unidades em tempo recorde, aprovação pela CONEP em tempo recorde, a logística com CT vacinas, fazendo diagnóstico em menos de 24 horas, os correios que foram mobilizados. Então, foi uma logística que eu nunca vi em território nacional. Então, um projeto que terminaria em 2, 3 anos, terminou em 4 meses. Esse foi o esforço e eu agradeço a acadêmica Patrícia Rocco pelo seu esforço. I would like to introduce Dr. Bruce Levine. Thank you, Bruce, for the opportunity to hear you. And Professor Bruce is a professor in cancer and gene therapy from the University of Pennsylvania School of Medicine. Dr. Levine's interest includes cell, gene, and biological therapy, clinical trials, development, manufacturing, and testing in lymphocyte growth, activation, and senescence. And he did his PhD at Johns Hopkins. I did my PhD also at Johns Hopkins, and I'm really very glad to see that we share the same education, the same in the same institution. And also, Dr. Levine has so many awards and honors that I will not mention all of them. And I just want to mention that he is the president of the International Society for Cell and Gene Therapy, and it's a good pleasure to hear you today talking about your specialty, and we are very glad to have you here. Thank you very much, Dr. Levine.
Well, thank you very much for the introduction and the invitation. I'm honored to be speaking for the Academy. Also very pleased to see my friends Patricia and Martin, albeit by video. I wish we were all together, but it's a different world. So I'm going to start sharing my screen, and let's share this. And let me know when you can see that. I'll go into presentation mode.
We can see and hear perfectly well.
Excellent. So I have a title that may be different in your program, but what I'm going to share with you is we believe a new pillar of medicine, using cells and engineered cells to treat disease. And first, by way of disclosure, I have a financial interest due to intellectual property and patents in the field of cell and gene therapy. That conflict is managed in accordance with the University of Pennsylvania policy and oversight. So Harold Varmus, in his Nobel Prize acceptance speech in 1989, described cancer in this way. He said, "We've only seen our monster more clearly," and described his scales and fangs in new ways that reveal a cancer cell to be a distorted version of our normal cells. And that is the problem, right? We know that cancers that succeed have evolved mechanisms of tumor immune evasion. They hide from the immune system, they suppress the immune system, they escape the immune system. And so because cancer is derived from our own cells, how can our immune system that evolved to tell self from non-self tell cancer? In some circumstances, this is relatively straightforward with viral-induced cancers or neoantigens, for example. But in truth, cancer-specific immune cells are very rare if they're present at all. And it's more like looking for a dull needle in a haystack. And so how can we get around this? How can we overcome the scarcity of tumor-specific immunity and tumor suppression? Well, the endogenous T cell receptor is a heterodimer that recognizes antigen presented by MHC, tissue type specific on the cell surface. These are intracellular antigens and account for roughly 2/3 of recognized cancer antigens. You can isolate tumor-specific T cell receptors, but they're still restricted by tissue type. So if you're thinking of treating a population, you need to have one of these T cell receptors for all tissue types. Chimeric antigen receptors are independent of MHC presentation. In that sense, they are off the shelf. They recognize surface antigens, and the CAR or chimera is essentially an antibody binding domain on the extracellular side of the cell and intracellular signaling moieties for T cells on the intracellular portion. Now that's a cartoon view, and I know you have heard or will be hearing from Martin also on immunotherapy, and I won't go into too much detail except to give you this picture of what a chimeric antigen receptor T cell attacking a tumor cell looks like. And in the course of 8 seconds, you're going to see recognition, activation, secretion of perforin and granzymes that were puncture holes in the cell membrane, and the tumor cell will explode. So here's recognition, and now you see that tumor cell exploding. Now, also recognize that this is a living drug. That CAR T cell survives and attacks the next tumor cell. It divides, and its daughter cells also attack tumor cells. So how we manufacture the cell therapy products is by collecting cells from a patient using a pheresis machine, so the same machine that is used to collect stem cells for blood stem cell transplant. So we get a bag of cells that is enriched for white cells. We can then isolate or deplete by centrifugal elucidation or magnetic selection. We're interested in the lymphocytes and specifically the T cells. We devised a way to activate T cells using an artificial dendritic cell that is a magnetic bead to which we conjugate two antibodies that engage receptors on the surface of the T cell. We use a lentivirus to deliver the gene, RNA that is reverse transcribed into DNA and permanently integrated into the genome. We expand the activated gene transduced cells in a bioreactor for several days, concentrate and wash, remove samples for quality control testing that address regulatory specifications for safety, purity, potency, and identity of the drug product. We formulate and cryopreserved, and then after testing, the cells can be released for infusion. And because we cryopreserve, these cells can be transshipped anywhere in the world. So I want to take you back 10 years to not our first CAR T cell trials, which were in HIV in the late 1990s, but our first CAR T cell trials in cancer, which initiated in 2010. The CAR T cells targeted CD19 present on B-cell leukemias and lymphomas. Our first 3 patients were all relapse refractory CLL patients with no other options. So this is Bill. Again, he is out of options with conventional treatments. In August 2010, his bone marrow was riddled with leukemia, all of the brown staining that you see. 6 months later, his bone marrow was clear. And in the first 3 patients that we treated, between 1.3 and 3.5 kilograms of leukemia was obliterated by the patient's own CAR T cells. So CLL is a relatively indolent disease. We wanted to next ask the question, when we move to a more rapidly growing malignancy, what would happen? And to answer that question, we opened up a clinical trial at Children's Hospital of Philadelphia in pediatric acute lymphoid leukemia, again, relapsed refractory patients. This is Emily, who was diagnosed in May 2010, had relapsed twice, came to Children's Hospital, received high-dose Cytoxan clofarabine, a regimen that would kill an adult, and only 1/3 of kids make it through. She did, yet in her bone marrow, she had 60% leukemic blast, lesions in her kidney, liver, and spleen, and she was not far from her demise. She received her CAR T cells in mid-April 2012. And I'm next going to show you a picture from May 2020. So Emily is doing fantastic. She's in high school, doing very well. Her parents started a philanthropic foundation, and they have become advocates for the field. Well, her clinical response was good news, but initially there was a very severe short-term toxicity, cytokine release syndrome, and we had observed this to a certain degree in our earlier 7 patients or adults that have been treated. But in tracking the cytokines and chemokines day by day, Emily's levels of IL-6 were 10 times higher than we had seen in any adult. She developed breathing difficulties, and blood pressure was extremely low. She was on 3 pressors, intubated, and in the intensive care unit, and not given a chance to come out of that until this data went to Carl June, our center director. And Carl's daughter has juvenile rheumatoid arthritis. He had been tracking FDA approved and in developed drugs for JRA and knew that there was a recently approved drug, tocilizumab, that blocked the IL-6 receptor. Communicated to the investigator at Children's Hospital. Fortunately, that drug was in the pharmacy. Emily received that and very rapidly recovered. And it is from Emily and subsequent patients that we've learned in most cases to manage this short-term toxicity. And I'll just remind you that the conventional treatments for childhood cancers carry significant long-term toxicities, which include hearing loss and other nervous system toxicities, cardiovascular toxicities, infertility, and pulmonary complications. And we have a short-term toxicity. Apart from B-cell aplasia, we don't have any other long-term toxicities that have been observed. And skipping ahead, we had an alliance with Novartis to develop this technology. They conducted 2 global pivotal clinical trials in pediatric ALL and diffuse large B-cell lymphoma, received FDA approval in August 2017, followed by EMA approval. Also, therapy developed by Kite Gilead, Yescarta, and Tecartus, approved in DLBCL and mantle cell lymphoma. Also by the FDA and the EMA. And Kymriah is also approved in Switzerland, Canada, the UK, Israel, Japan, and Australia. I know that there are clinical trials in Brazil. I visited a manufacturing facility there and look forward to that developing further. This is a long-term therapy, again, coming back to the durability. 1 infusion lasts years. Here are the 2 of the first 3 patients who entered complete remission. We can track their CAR T cells and the red triangles out to 7 years, shown here as reported in 2018. And I'm pleased to say, as of August 2020, our first patient celebrated his 10-year T-cell versary, if you will, and we observed and detected CAR T cells in his peripheral blood. So we are in the era of synthetic biology where we can design logic-gated Boolean CARs to function in an and or not fashion, checkpoint-resistant CARs, safety switches, and engineer conditional or stealth CARs. And we need all of these in solid tumors for a number of reasons. And that is that solid tumors include a number of challenges that are generally not observed in blood cell cancers. Difficulties in antigen coverage, antigen specificity, antigen escape, a much more immunosuppressive microenvironment, as well as the challenges of trafficking, particularly in pancreatic cancer, which is has highly stromal aspects. So I'd like to give you some very quick snapshots of a couple clinical trials that we have ongoing. The first is in prostate cancer, where we're targeting prostate-specific membrane antigen. Now, in the US, we have 175,000 cases per year. I imagine similar numbers in Brazil. The challenge is that 20% of those patients develop metastatic castrate-resistant prostate cancer within 5 years of diagnosis. Another challenging thing is that prostate cancer cells, again, very immunosuppressive through secreting high levels of TGF-beta. So how to approach this? Well, we engineer a decoy receptor, a dominant negative TGF-beta receptor to the CAR construct. And the design of that is shown here. And normally, prostate cancer cells secreting TGF-beta engages the wild-type TGF-beta receptor on the surface of a T cell, transmits the signal, and that T cell is suppressed. Now, with the dominant negative TGF-beta in this construct with the chimeric antigen receptor to recognize PSMA, that TGF-beta can come in, but the signal is blocked, making these CAR T cells immune to those immunosuppressive effects. So we have opened up a clinical trial at the University of Pennsylvania. We have treated 3 cohorts of patients here and in dose escalation with the integration at the next level dose with preconditioning chemotherapy. And for comparison on in vivo engraftment, I'm showing you selected patients from our CD19 CAR T cell trials, and you can see when we go to this higher dose, we have much higher level of engraftment, and I've superimposed that here on the right. You can see what a difference that is. Now, this particular patient was a 72-year-old male who had previously failed surgery, radiation, and 4 lines of standard therapy. He had low disease burden, and you can see what happened to his PSMA here. It dropped to undetectable just over 2 weeks after infusion. He did develop cytokine release syndrome that was resolving. However, at that point, due to leukopenia and neutropenia, he developed grade 4 cytokine release syndrome and unfortunately developed enterococcal septicemia and passed away due to that. But we have treated additional subjects, and we're seeing similar trends with the PSA. So 12 patients across 5 cohorts and 2 separate clinical trials. Our goal is to identify a phase 2 dose before the end of the year and commence a phase 2 trial in 2021. And so the next trial is targeting a glycoepitope, so-called TN-MUC1. And this stumpy MUC1 is present in a number of solid cancers, including non-small cell lung cancer, pancreatic cancer, triple negative breast cancer, ovarian cancer, and multiple myeloma. And finally, just very briefly, touching on our Crispr trial that was published in February. This was the first Crispr trial conducted outside of China. And what we did in this trial was to knock out the endogenous receptor, knock out PD-1, the checkpoint receptor, and deliver a T-cell receptor targeting NY-ESO-1, a cancer testis antigen that is present in multiple myeloma, synovial sarcoma, and a number of other cancers. Because this is published, and in the interest of time, I won't review the data, but just review the summary of the findings. We proved that a very difficult technology can be manufactured. We showed safety and feasibility. We did not observe cytokine release syndrome or serious adverse events. The best clinical response was stable disease. We observed persistent engraftment of the edited cells for at least 6 months. There had been concerns about the potential immunogenicity of Cas9 due to originating from bacteria, but we did not observe that. And up to 10% of the T-cell receptor modified cells have disruption of the checkpoint receptor. And at the single cell level, we were able to demonstrate digenic and trigenic editing. Important to note that we conducted this clinical trial with 2016, 2017 gene editing technology. You all appreciate, I'm sure, the rapid pace of evolution in CRISPR, and our next trial that will open next year is incorporating the latest technology. And last issue I want to touch on before closing is responsibility and ethical considerations in cell and gene therapy. And this is a cartoon that crossed my desk, a mother wheeling a baby in a stroller, and an older lady passing by says, "What a beautiful baby." And the mother says, "We can thank the man upstairs." Well, who is the man upstairs? That's at Genomicorp gene-edited babies. And I think you know the event to which I'm referring that occurred in China. And the bioethicist Art Kaplan termed this stupendously immoral renegade science. And I'll add that it was a technical disaster as well. So separating hope from hype is the proliferation of these so-called stem cell clinics that sell direct to consumer, marketing cures for everything from Alzheimer's to autism to arthritis. And this is of a concern due to a loophole in regulations. They have escaped FDA enforcement that is ending in May of 2021. There have also been court cases against some of these predatory clinics, and there are also cell banks, T-cell banks, tooth banks that advertise direct to consumer to cure all diseases. So we have a presidential task force at the International Society for Cell and Gene Therapy to define this. I won't go through all of these criteria. This is meant more for public consumption, but just to advocate for communicating to the public as understanding the difference between scientifically validated and clinically rigorous studies in the development of new technology. So we do have threats. We have bad actors co-opting new technology terms. We have lax enforcement, ethics dumping, and regulatory brokerage. We have public confusion on new technology. How many of your neighbors, friends can explain CRISPR to you? Moore's law in biology is the technology is evolving faster than the public can understand, and the regulations and guidances and the drug industry evolved in an era of small molecules and biologics. So what do we need for wider patient access, improvements in manufacturing, incorporation of automation to enhance potency, education and training at all levels, and recognize that these are financially complex drugs. So we need to think of payment mechanisms that link price to outcome, incorporating predictive biomarkers. And finally, I showed you a picture of Emily. On the left is Sophia, who responded initially, but unfortunately relapsed. So we have to keep in mind developing improvements so that we have wider access and the ability to treat all patients. Acknowledging our collaborators and funders, it truly does take a village to develop these therapies. And I thank you for your attention and look forward to discussion.
Thank you very much for the excellent talk.
Go
ahead, Marcelo.
Thank you. Thank you, Mr. President, and thank you, Dr. Levine, for this impressive presentation that shows the possibility of the frontier of science in the treatment of cancer in this presentation. You are showing the importance of the continuous investment in science, but not only that, but the importance of articulation of scientists and scientific regulatory agencies in the different countries and science foundations. You mentioned the validation of the studies and all the procedures to put all this technology to improve the life of our population. Thank you very much for this brilliant presentation. We will have a space for debate, but the next presentation will be Dr. Marun Khoury, and I would like to thank you, Marun, for this possibility to have you here. Dr. Marun had his PhD in gene therapy at the University of Montpellier in France, postdoc at MIT, and he's a professor at University de Los Angeles, em Santiago do Chile, e é diretor do Laboratório de Investigação de Terapias de Células Madres e professor associado full-time e também vice-presidente internacional da Sociedade de Cell Terapia de Latinoamérica. Gracias por la posibilidad de estar con nosotros. Thank you.
Sim, muito obrigado pelo convite. Eu me sinto muito honrado em participar neste simpósio. So I'm the ex-vice president, the current vice president of regional ISCT for South and Central is Professor Patricia Rocco, and she's doing a great job. So I'm happy to participate, and let me share with you my screen.
In the meantime, let me thank you very much on behalf of the Academy for your presence here among us. I was going to speak in my Portuñol, but I'm afraid that yours is better than mine. So I switch it to English. Thank you very much for coming.
Thank you again. So of course, I would have been even happier to be with you in Brasilia, but I think we can fix that with Bruce. So I'll be talking to you. So I'll be talking to you about different cellular strategies for controlling and even restricting the inflammation, preventing it from tissue destruction and injuries. So by cellular strategies, I'm going to focus mostly on mesenchymal stem cells that has been used widely for treating autoimmune and other inflammatory diseases. So this is my second virtual meeting that I present with the presence of Bruce. With Bruce, we have tried to compensate the lack of social time during breaks and after the talks to create this kind of an avatar where we sent our avatar to the ISCT annual meeting that was held in Paris this year, and this will not be an exception. I will be as well sending our avatars to Brasilia, and then we'll be very happy to be there with you. So since again this is a virtual meeting, I would like to invite you to a virtual campus tour. This is the Faculty of Medicine. So our university lies on the foot of the Los Andes Mountains. And at the corner, you can see the clinical hospital where we are currently doing our mesenchymal stem cell based trials in different diseases, including lupus and osteoarthritis. And just behind the lab, we have our laboratory where I'm standing today with my team, where we're doing the different investigation regarding the use of these cells. So this is said, there are more than 100 different types of autoimmune disorders. And then currently there are about 126 trials that uses MSCs for autoimmune disease. Most of them are based in the US and Europe, but as well, there are a couple of these trials in our region. And our group is starting a phase 2 clinical study in lupus that I will be mentioning as well. So I would like to use the timeline of the discovery of MSC to tell you when the MSC and the immune regulation started. So after the discovery and characterization of these cells, the first paper mentioning the immunosuppressive effect of MSCs was in 2002. And where they were able to transplant skin to a baboon. At 2003, I started my PhD in France, and I was sharing, the offices were very small for students, and I was sharing my office with Farida Jawad, who actually published in the same year the first paper showing that MSC immunosuppressive effect might favors a tumor growth in mice. Following to that, in 2004, Katrina LeBlanc in Sweden showed the first case report of the use of MSC in acute graft versus whole disease. Following this, there was a spike of using these cells in different trials, including Crohn's disease, multiple sclerosis, whether it was autologous or allogeneic. And in 2009, before I moved to Chile, Fernando Figueroa in the same campus did the first autologous treatment of MSC in lupus patients. This was not successful, and this is why it took us 10 more years to come back with a defined product. So beyond these different trials, just very recently in 2018, EMA, the European Medical Agency, approved the first drug based on mesenchymal stem cell called alofisel for the treatment of Crohn's disease. So the reason why we use these cells for autoimmune diseases or or in an allogeneic use is because they express they do not express MHC2, and they express low TCR costimulatory molecule. You can think about it as a invisibility cloak where you can inject them there. They can not be seen by the immune cells until something happens, until they react with something, and then they start expressing these factors. This is why people call them as hypoimmunogenic. Other people label them as immunoprivileged, but I would like mostly to use the term immunoevasive because they can evade the immune system to a certain point, and then they can be discovered and then cleared away. So this is we're talking about a global immunosuppressive effect because these cells are able to interact with all immune cells and their subtype, leading to different outcomes and different mechanisms. What I mentioned was the MSC, the naive, but you have to understand that mesenchymal stem cells have a yin and yang, and by this I mean the polarization according to a pre-stimulation step that can be done through poly-IC or LPS incubation that can lead to two phases. One is anti-inflammatory, and the other one is pro-inflammatory. This is not good and bad. This is whether you want an anti-inflammatory effect for example, autoimmune diseases, or you want a pro-inflammatory effect, for example, for the treatment of cancer. So, and recently, last month in our group, our PhD student, Monica, published a paper showing that within one stimulation, this means only with LPS, depending on the exposure time, she can have a pro-inflammatory or an anti-inflammatory. In that case, when LPS is put for 1 hour, we have a pro, and when we have for a longer time, 24 and 48 hours, we can get an anti-inflammatory effect. From this, she showed that the immunosuppression is as well modulated. In green here, we have the naive here, MSCs in black, and then whether the cells are pro or anti, they can induce or suppress greatly the T cells in vitro. And then as well, these cells were injected into a EAE model, mice model, where it's been shown that MSC anti-inflammatory, so with LPS 48 hour, we have obtained a higher therapeutic potential. And on the contrary, when you're injecting pro-inflammatory induced MSC, you can have even aggravation of the disease. So this means the failure or the success of MSC-based therapy is commanded by their inflammatory status, or even better said, phenotype. So this as well opened a space for pre-treatment for enhancing these cells through for their immunomodulatory effect. And there are multiple ways of enhancing them, whether genetically or non-genetically. In our group, led by Patricia Luce-Crawford, she just showed that you can do a metabolic reprogramming. And what she showed by doing driving them through more a glycolytic metabolism, she can augment, she can increase their immunosuppression capacity. And this work was done through the use of ATP synthase inhibition, and that was published just recently this months. So you can reprogram them metabolically and show that they effect and therapeutic effect can be enhanced. Following on this, as different speaker this morning mentioned, the cell therapy has multiple limitation. Professor Rocco told about embolia, etc., and the reason why she wants to move to an exosome strategy. They as well have other limitation, including low homing and very high clearance. So it's very important to start looking at the mechanism of action. Think about a plane that crashed, and then you were able to get that black box. The only thing you want to know is why it crashed and what was the last discussion between the pilot and the co-pilot. If we can do that while injecting MSC, then we can decipher why it's cleared very early and how we can improve their role. And this is exactly what we are trying to do. So MSC are known to have to be able to differentiate to different tissue, and their immunomodulatory property comes from mainly secreted paracrine factors. I'm talking about soluble factors, but as well exosome, delivering packages of RNA and protein, and just recently transferring their own mitochondria. And this is going to be the focus of my talk. So I'm going to take you back to your first biology course when you heard about the endosymbiotic theory, where mitochondria used to be a small bacteria hovering around, and then they met a large bacteria called the eukaryotic cells, and symbiosis begins. Then cooperative living started, and then they actually removed a couple of their genome, and then they were happy ever after. So the comeback of this story is that they never forget their freedom, and then they might want to again go and browse around in our body. So what I'm saying here is that mitochondria concept from being only power plant to immunomodulatory forces in cell therapy is exactly what I will be detailing. In a paper was published in 2013 in Nature, showing that when you have a stroke, dendritic cells transfer, astrocyte cells transfer their mitochondria to damage neurons spontaneously. This is without any intervention. Then following this paper, many researchers showed that the injection of mesenchymal stem cells in lung injury models or other different model are able to actually transfer their mitochondria to immune cells, including macrophage, leading to a suppression of the inflammation. And this as well has been shown in heart injury, but as well in the bone marrow niche between mesenchymal stem cell and HSC. So how does this happen? So myRON, one of the protein responsible of intracellular mitochondria and intercellular mitochondrial dynamic, we have the creation of what we see here, the nanotubules, and through the nanotubule we have mitochondria being transferred from a donor to a receptor cell. This was demonstrated in a Embryo Journal in 2014. And then TNT are able not only to channel mitochondria, but as well extracellular, intracellular, lysosomal, and even Golgi vesicles from one to another cell. And what we're doing right now in our lab is understanding the mechanism of this transfer from the MSC. And this is the work of our PhD student, Francesca Velarde, and she's trying to decipher other mechanisms, including macrovesicle, where mitochondria have been shown to be able to be engulfed in these vesicles and then being shuttled to other cells, among other mechanisms, including gap junction, apoptotic body, and cell fusion. So the first in vivo mitochondrial transfer, I will refer it as MitoT, happened in a heart ischemia model by James McCulley, where he injected directly isolated mitochondria and looked aside for functional recovery after injecting those mitochondria, which he showed that this recovery occurs in heart injected with mitochondria. In red dots here, we are able to see the mitochondria within the heart, However, he was not able to see internalization of these mitochondria by cardiomyocyte. Therefore, the mechanism is still to be deciphered. So what is our focus? And coming back to autoimmune and inflammation, the reasonable question is that taking into consideration that the engagement of specific metabolic pathway is associated with cell differentiation and function in lymphoid cells. Therefore, question is, do MSCs shape the immune response by transferring this mitochondria? So one first essay that we started, so before doing those assays, it was known that MSCs in an in vivo context were able to transfer their mitochondria to macrophages and induce a polarization toward a anti-inflammatory status. But there was no information regarding lymphocytes. So what we did is we co-cultured PBMCs with MSC that we stain for their mitochondria. And what we were able to show that MSC, not only from umbilical cord, but as well from other origin, including menstrual and bone marrow, are able to transfer their mitochondria to lymphocytes. And I'm talking about, of course, B and T, but as well to other type at different levels. Here we're averaging about 40% of lymphocytes receiving mitochondria from MSCs. And when we're looking at subtypes, we can see that CD4 positive cells are more permissive to receive these cells. We're talking about here healthy cells so far. So how does that occur under the microscope? The big, huge cells here, Star Wars cells look like is an MSC. And what we have is a small vessels. Those are the lymphocyte that comes, and with an action of touch and go, take the mitochondria and leave. And then we have a circular effect of these mitochondria. What we are able to see here is a Cd3 staining of lymphocyte, and the green dots are mesenchymal stem mitochondria within the lymphocytes, all right? So this is a mechanism that occurs very fast. At 3 hours, we're able to get about 30% of positive cells that acquire those mitochondria. And the following question is whether these transfer can cross barriers, and if we have higher or lower transfer when the lymphocytes comes from a disease, from an autoimmune disease. So what we were able to see that to show that the human cells can be transferred through the same co-culture system to mouse lymphocytes. And more interestingly, here we're looking at lupus mice model, where we're able to see a dramatic increase in the uptake. So when we have healthy mice, we still have a transfer which is less efficient than human cells. But then when we co-culture them with lupic mice cells, we have an important increase of the transfer, all right? And of course, we have used human genes and mouse SNPs to identify that there was no fusion between the human and the mouse cells. So everything I showed you till now was the co-culture of the cells. The second experiment was to isolate mitochondria from the mesenchymal stem cell and spray them through a protocol we call mitoception to over the PBMCs. And the reason behind this experiment is we want to see the effect of the mitochondria alone without the effect of other factors such as the soluble and vesicle that can be as well released from MSC. So we want to have a clear system to assay the effect of the transfer of mitochondria to other cells. So we were glad to show that we were able to get about the same average of mitoception or mitotransfer to these cells as seen in the co-culture experiment. And what I'm showing you here is a confocal microscopy where red dots are the endogenous lymphocyte mitochondria, whereas the green dots are the mitochondria that were transferred, isolated, and then transferred from mesenchymal stem cell. Having a quantification of these mitochondria, we were able to show that the biomass of mitochondria in lymphocyte increased dramatically. We wanted to know if these mitochondria that were transferred are functional. We did multiple assays, including measuring ROS, looking at TMRM, which is the membrane potential of these mitochondria, and as well as the ATP content of the isolated mitochondria, and we were able to show that they were functional. We as well noticed a metabolic change through oxygen consumption rate and ECAR. And then we wanted to know what is the effect, what is the functional effect, if they are able to determine the fate of these naive lymphocytes that acquired these mitochondria through a RNA-seq analysis. We're able to see different differentially expressed genes, and then we look at different signaling pathway. And here we're looking at the combined score and the adjusted p-value, where the most important pathway they were changed or modulated were the inflammatory response and the regulation of T-cell activation as well. So with this, we came up with a pathway analysis that led us to the following findings, where we have seen functional effect of mitotransfer by driving spontaneous differentiation of CD4 positive T lymphocyte toward a Treg, as seen here. This is the lymphocyte that have not received the mitochondria, or they were resistant to mitochondrial transfer, whereas the one that received it start forming cluster, and this cluster happened to be CD25 positive and FOXP3 positive, therefore Tregs. And then the second question, we wanted to know if these spontaneous fate induction were delivering us with functional Tregs for obvious translational interest. And then doing a proliferation assay where we co-culture these de novo Tregs with PBMCs, we were able to show that they are able to have a immunosuppressive and functional effect without any additional stimulus. So following question is to know whether these MSC that are derived, the mitochondria derived from MSC, are specific to mesenchymal stem cells, or we can use any cells. So when doing a transfer from fibroblasts or from other PBMCs, the effect was not seen, and not only that, the transfer was, or the occurrence of the CD25 and FOXP3 cells were as low at 2 and 3% compared to 20 and 30% when we used MSC's mitochondria. Second functional effect, we wonder whether we can confer a anti-aging, or better said, having an anti-apoptotic or anti-aging, immunoaging effect. So for this, we have looked at the apoptotic pathway, and then we were able to see that within the lymphocytes that received mitochondria, we have an increase of different factors, including BCL2. So we induce cell death through, or apoptosis through staurosporin, which is a wide antibiotics. And then to our surprise, we're able to see that the lymphocyte that received mitochondria from MSCs are more resistant to STS-induced apoptosis than the one that did not receive. We wanted to decipher as well the mechanism, and we were able to show that the caspase-3 activation is prevented on mitocepted T lymphocyte compared to non-mitocepted cells, conferring this kind of anti-protective effect or anti-apoptosis effect. Taking all these experiments to in vivo, we wanted to see the impact of this transfer on immune tissue damaging in a xenogenic model of graft versus host disease. So what we did is we now injected those PBMCs that were mitocepted, so that contain a human mitochondrial MSC, and compare them with the one that did not see in their mitochondrial biomass augmented. And what we're able to see is that the transfer of mitocepted PBMCs was associated with clinical improvement, but as well increased survival and reduced damage of the immune tissue infiltration compared to disease induction with untreated PBMCs. Looking at the repopulation of human cells in the mice, we've seen a decrease of PBMC that received the mitochondria, but as well a decrease of CD8 and CD4 positive T cells. And then we were able to observe a important decrease of the gravity of the clinical score of different tissue, including small intestine, liver, and of course lung, and then less T-cell infiltration and the lavage of these tissue. So the conclusion here is that mitotransfer spontaneously induced Treg immune function reprogramming, and these can lead to a regulation of the inflammation and control of tissue damage. This marked therapeutic effect might pave the way for future organelle-based therapy or cell-free therapy. This means only using a part of the cells, in this case mitochondria, versus injection of the whole MSC. So going back to the mechanism of action, what we were able to show here, beside what was known with the macrophage polarization, we're able to show that immunomodulation occurs not only by MSC, but as well through the mitochondria transfer alone or in association with other factor inducing a Treg phenotype, but as well an anti-apoptotic and an oxidative stress regulation. So left and right brain conclusion, there are many questions to be asked as scholar, but as well as clinician, how we can use this information toward the clinic by enhancing the mesenchymal product through preconditioning or through metabolic modification or through using their byproducts such as mitochondria and exosome. Clinician has as well multiple questions still to answer to be able to deliver treatment for refractory autoimmune diseases. And then we wanted to use this information to immediately through an ongoing clinical trial that we have initiated in last year. That was initiated by Fernando Figueroa from our department, where we're injecting mesenchymal stem cell. And the idea here was to see if we can see the evidence of mitotransfer in patient, knowing that the mesenchymal stem cell that we are injecting coming from a different donor, we're able to find the specific SNPs of their mitochondria, and this is what we're doing. On the other side, just recently it's been shown that mitochondria, not only their DNA and actually mitochondrial DNA is implicated in multiple autoimmune diseases, but now we're talking about functional mitochondria circulating, what we call them this circulating cell-free mitochondria, whether they are free or encapsulated into vesicles. And this has been shown to occur in healthy individual. What we wanted to see is from our patient, treated and not untreated, whether these cell-free mitochondria are an important marker of the disease progression or of the treatment efficacy. So what we did here is and this was done by Yesia Hidalgo in my group, that actually purified mitochondria from the plasma. She looked at their fitness or their potential through flow cytometry. And I'm showing you this unpublished data. This is a 2-day old out of the oven data. And when, of course, this is very preliminary, but then what she was able to show is not only they look fit through different markers, but as well, she took those mitochondria from the plasma of patient, incubate them with PBMCs in the same protocol we use with the mitochondria from mesenchymal stem cell, 24-hour culture. And the good surprise is that these mitochondria were actually internalized into these PBMCs. And of course, we will have to define what is the function of these cell-free circulating and how we can use them to as a biomarker or even as therapeutical agents. With this, I would like to thank, of course, the whole team in my group, researcher, the medical group, the cell production group, and as well the administration and the funding we were able to receive with the support of our university. And with this, I would like to thank you very much.
Muchas gracias por la charla que fue excelente. Y yo quiero ahora invitar al doctor Marcello Barcinski para seguir con el programa.
Muito obrigado, senhor presidente. Então, enfim, agradeço as excelentes contribuições dos dois conferencistas, Dr. Levine, Dr. Curi. Dr. Levine, thank you very much for your excellent talk. Dr. Curi, thank you very much also for your exciting presentation. And both of the presentations are open for questions and discussion. So please, the one that is Martin Bonamino, then Patricia Rocco, and please, Martin.
Okay, so thank you, Marcelo, and I have a quick question for each one of you, Marion and Bruce. So Bruce, hi, nice to see you here again. So it's quite in the same line for both of you, because there's a preprint that has just come out a few days ago showing, Bruce probably is aware of this, the transfer of telomeres from dendritic cells to T cells during the synapse. So this elongates the telomeres from the T cells and make them more resistant to telomere erosion during expansion. So this is a question that's combined for both of you. How do you envision this being applied? If this could be something feasible to produce in a clinical grade setting? And to Maroon, especially for this very interesting and provocative piece of data on the mitochondrial transfer. So you have been transferring this MSC from these more immune regulatory phenotype cells, right? But you mentioned it at the beginning of your talk that you can modulate this function to be more pro-inflammatory. So have you tried to check whether those mitochondria will bring a different phenotype, and whether this could be used to enhance, for instance, anti-tumor or antiviral functions from T cells?
Well, we can do the discussion in both ways, or we ask all the questions and then you answer all of them, or you can answer one by one, which you prefer.
Well, perhaps I'll answer part of Martin's and turn it over to Maroon, and we can go from there, if that's okay.
Okay, fine. Please,
So,
Bruce.
Thank you, Martin, for the questions. And, you know, I was thinking of that telomere paper in the back of my mind as Maroon was presenting his mitotransfer and the work of exosomes. It's amazing how much we learn, right? We didn't know RNA was a catalytic molecule until a decade or two ago, and now the importance of all of these subcellular and molecular components being transferred among cells. So, you know, we showed a number of years ago that our method of T cell stimulation using an artificial antigen presenting cell that is a bead to which we conjugate anti-CD3 and anti-CD28 antibodies induces telomerase, which in fact, upon repetitive stimulation, stabilizes the length of telomeres. We also showed that there's a correlation between longer telomeres in naive T cells compared to shorter telomeres in memory T cells, and that telomeres shorten with age in T cells. So in a sense, it's not surprising, but it's really cool to know that T cells have this power to co-opt telomeres from dendritic cells. And I think we will find a way to use that, if not therapeutically, then certainly in preclinical development and discovery. And the link to Maroon's work is that we also know that there is a very strong link between T cell metabolism and T cell fitness. And that central memory T cells, which are the quality T cells that we look in developing a therapeutic product, are high in oxidative phosphorylation, and that the exhausted terminal effector cells, or even during activation, there is by necessity a switch to glycolysis. So I'm thinking, as Maroon is speaking, how can we exploit this knowledge? How can we incorporate it? It's extremely interesting biologically, but I think it also has therapeutic implications. So Maroon, I'll turn over to you.
Thank you very much, and I agree with you, Bruce. Martin, I got scared by your question just because yesterday or the day before I was discussing this with my student. Actually, she graduated, so now she's a postdoc. And then we decided to write a hypothesis review about the transfer of different subtypes. So this is one of her tasks. So I wonder if you have some cameras around me. However, to answer, I can answer only one part of your question. We try to actually drive them to an anti-inflammatory status and then do the transfer. We didn't see any difference in terms of immediate percentage of mito transfer or even the emergence of Tregs, but we will have to do it as well with the pro-inflammatory, pro-inflammatory phenotype and see whether we are able to see a difference in the downstream functions.
Thank you very much. Patricia Rocco, please.
First of all, I'd like to thank Bruce for being here. Bruce is now the International Society of Cell Therapy President, and I'm very honored that you are here in the Brazilian Academy of Medicine. I'm very happy also that Maroon is here. We have been working in the last several, I think 5 years, and I'd like to do some general questions in order to. International Society of Cell Therapy. I think something is open. Oh, now it's okay. Yes, they closed. And I would like to ask Bruce as well as Maroon, based on all talks that we have during the afternoon. So for Maroon, I think that in the academy, it's important to know whether or not the use of mitochondria as therapy is as good as, for instance, extracellular vesicles or mesenchymal stromal cells, because you showed us that how important it is to treat some disease with the mitochondria and the possible mechanism of action. But we also know that there are several papers showing the advantage of using mitochondrial therapy compared to the cells per se or the vesicles that can be produced by. So I'd like your opinion regarding this issue. And for Bruce, Martin presented us some data related to CAR T cells. And he showed us the cost. And we know that there are some pros and cons and for using CAR T cells. But one important limitation is the cost. And Martin showed us that they are trying to change, to do some changes in order to reduce this cost. I'd like your opinion regarding what are you doing indeed in order to reduce the cost of this therapy, you know, for leukemia in the case that Martin showed us. So I don't know who will start.
I'll defer to Maroon on this one.
Perhaps Maroon and then Bruce. Maroon, please.
Okay. Okay. So thank you, Patricia. And we've been collaborating as well on different projects, including mitochondrial transfer. So there are two strategies here to be considered, whether we want to use mitochondria as a sole therapy. And there are a clinical trial that is ongoing in the US and I think Harvard, injecting mitochondria from autologous tissues. This is the acellular product it would be, but as well, there is another strategy by augmenting the mitochondrial content, whether T cells could be for CAR T, making them more energetic. Of course, we don't want them to turn into Tregs, but then we have to fine tune what type of mitochondria we want to transfer and then make them more robust. So we can have a strategy by using mitochondria alone, or we can make cells that pre-existing, whether there are mesenchymal stem cells. And there is a paper, and we have preliminary data where we showed that we can increase the biomass of mesenchymal stem cell. This means mitocepting mitochondria to the mesenchymal stem cell from their sister cells, and then they can increase their effect of donation. I mean, charity depends on how much money you have in your pocket. If the mechanism is giving away their mitochondria, then you better have a lot of those mitochondria. So there are two strategies, and I like the combination of these strategies to actually go where MSC alone, naive MSC or exosome alone are not doing that great yet, and then we can combine them with a boost, let's say, of mitochondria. And of course, we'll have to do a neck-to-neck comparison to see if mitochondria alone versus MSC alone are having the same effect, and what are the pros and cons of using one or the other in terms of manufacturing. Same question we've been asking to cell therapy can apply to the future organelle therapy.
Thank you.
Thank you. Bruce, please.
Yes, so thank you, Patricia, for the important question. And Martin has done some important work in this area. I'll answer in a number of ways that we and others are thinking about this. We have a very labor-intensive process. We need to automate it. We have a process that in general for the commercial products is a 9 or 10 day manufacturing in the lab. We know we can shorten that. We have validated for clinical manufacturing a 3-day process. And in 2021, we're going to open that trial. So that is one way of reducing the cost. But recall that these are durable therapies. And if we can think of them as taking the place of some conventional therapies and reducing the long-term side effects and the effect on caregivers and the effect on lost work, then the equation does not look as intensive as first viewed. I'll also add that we're looking at biomarkers of prediction of who would respond, and then you can segregate those patients that are more likely to benefit. And then you're actually improving the value because you're channeling the patients who would not benefit to alternative therapies. And finally, I'll answer that we're working on ways to improve the potency of cells, reduction in the ancillary materials, improvements in the method of gene delivery that will reduce costs. And in the end, if you think of new technologies, the first automobiles, the first cell phones.
Oh, yeah, absolutely.
Yes, the first generations are expensive and clunky. I think that's where we are. But part of the ripple effect of findings of clinical successes, we now have investments in technology and reagents and equipment that are vastly accelerating the rate of discovery and development. And I think in the coming few years, we're going to see improvements that will reduce costs and improve access. And on a personal note, I'm on the scientific advisory board of a company in India looking to bring CAR T cells to India. Also, costs are a consideration. So we're working on a global basis and very optimistic about the future.
Thank you, Bruce.
Thank you
so
very
much.
So Dr. José Suassuna wants to make some questions. Please, Suassuna. Put on your microphone. Yeah, please.
I'm muting my microphone. Many thanks for 2 wonderful lectures. And what really surprised me was to know about how mitochondria can traffic from one place to another. But I was wondering about the mechanisms that that process of uptake from one cell happens. Because, I mean, if you connect 2 cells, why the traffic goes in one direction and not the reverse one? So there must be some clues on one side or another to make the mitochondria follow that particular direction. And what really strikes me the most is how free mitochondria would be able to enter a cell. Is it by the an active process, perhaps by the bacteria ancestry, or is it something that the host cell would do to engulf those mitochondria?
Yes, Thank you, Jose. Those are excellent questions, and some of them are being answered experimentally by us. So what I showed you is a unidirectional transfer from MSCs towards lymphocytes. We have assayed for bidirectional, and we haven't seen transfer from lymphocyte towards MSC because we were using probably healthy. There was a paper published 2 years ago where they show when you're putting cancer cells, I wasn't sure what type of cancer they used, but anyways, what they seen is that the lymphocytes were propelling their defective mitochondria outside. Mesenchymal stem cells were able to sense those mitochondria. So the mitochondrial transfer was bidirectional in that case, and then transfer the healthy ones. In our case, we use healthy versus healthy, and we haven't seen this. However, when we did a co-culture between MSCs and chondrocyte for a different project, we were able to see those two highways where we have mitochondria going from one cell to another and vice versa. The TNT, you have to understand, it has an anchoring point. So the TNT is a prolongation that goes from one cell to another, most probably to channel their mitochondria. And then it can as well receive a TNT towards the same cells and anchor it towards it. And then we can have both ways. I'm not sure if within some same TNT, we can have mitochondria going that way and then the other way, the other one going in the opposite way. But this is something we're doing right now to understand if it is bidirection within the same TNT, or you need to have two different TNT. This is something that we are studying. Other mechanism than TNT as well has been described, including different factors, CD38, different molecule being able to uptake those cells, and then of course this brings us to think that that the uptake might be similar to their ancestry, which are bacterial. In our case, in the mitoception, we were not able. We used different inhibition. We did macropinocytosis inhibitors. We tried to inhibit different pathway, and we were not able to completely block this uptake. So there are something going on that we still need to understand.
Thank you, Maroon. I want to ask if somebody else wants to make comments or questions. If not, I want to make a question to Maroon. It has to do with the previous question, and the generation of Tregs is usually by mitochondrial transfer from MSC, or you need some kind of inflammatory or anti-inflammatory stress for this, for the appearance of Tregs dependent on the mitochondrial transfers.
Yes. So the differentiation toward Tregs is usually done by a cocktail of cytokine. Then there are clinical protocol isolating cells and then inducing them toward Tregs. So these are defined protocol using concentration of cytokines such as IL-2, etc. In our case, this was a surprising fact. So I will not say this is now a gold standard by any mean, but I think this could be combined to the clinical protocol that are currently being used for the generation of Tregs. This could be an enhancement of a protocol that is not working correctly by actually adding mitochondria. So I see it as a combination. But again, what I showed was a kind of a surprising effect rather than a gold standard.
Thank you very much. Dr. Aderbal Sabra, please.
Yeah, well, very interesting talks. Very nice opportunity to keep up with the ideas. The core problem in COVID-19 is inflammation. We have been talking about inflammation. How long or how far we are from a mitochondria product in the market? Or have you been using your experience, Dr. Maroon, using mitochondria inflammations to regulate the depression of T cells in COVID-19, using the stimulation of Tregs or T cells with the mitochondria? And in any case, have you been done this?
Okay. Thank you for your question. So first, we're not in a hurry to go for the clinic because we have to see and learn from the cell therapy and all the limitation and try to first overcome these limitations, whether it's cost, manufacturing, efficacy, etc. As I mentioned, there is an ongoing clinical trial in the US using mitochondria for cardiomyopathy. There is no result yet, but at least we know that now it's something close to the clinic. For your second question, we have filed a grant application with the Professor Rocco. And the reason is that some protein of COVID, ORFs, are able to hijack the mitochondria of target cells. There is a protein called MAVS for mitochondrial antiviral protein, and that protein can activate interferon response. So we wanted to kind of counteract the hijacking of COVID of these of this protein, making the mitochondria elongated and therefore defective by supplementing mitochondria and see if we can do the balance. Unfortunately, we were not granted that proposal, but then the idea is still there, and most probably we will do it one way or another, as always, with the collaboration we have with the University of Rio. But yeah.
Thank you.
Thank you, Maroon. Martin Bonamino, please.
Bruce, Marcelo asked me before, earlier today, about the pricing, and Patricia already mentioned this again. And I was commenting that maybe you can further extend this on that these therapies are very disruptive in terms of pricing, but also in terms of reimbursement and ways of paying for them. So maybe you could make some considerations on the different types of reimbursement that are being proposed and practiced for not only for CAR T cells, but for other advanced therapies, as if you want to call them this way.
Sure. Well, I'm trained as an immunologist, but I'll do my best based on what I've been hearing. And in the U.S., it's complicated because we don't have a national healthcare system. We have a series of third-party payers, and there's the concern because insurance is tied to employer. If a patient moves from one employer to another, the insurance is different, and who is likely or less likely to cover. So there has to be the addressing of the incentives to reimburse. Now, one thing I mentioned is very briefly is value-based payment, and that essentially is pay for performance. So if a patient responds within 3 months, then there's reimbursement. But if there is no response, then there is not reimbursement. So that's an incentive on the developers to bring their most potent and effective therapies forward for approval. Also in the US, it's complicated because the regulatory approval is not tied to the pricing. There's a different agency that does the pricing. Another proposal that has been discussed is annuity payments. So there would be reimbursements of every period, whether you pick 6 months or a year, and there would be reimbursement payments periodically depending on the response of the patient. So some of those have been discussed. I think it's more likely to have alternative reimbursement methods adopted in a country outside of the US because of our historical complexity. But also come back to part of the answer that I gave to Patricia before, which is it may be faster to make improvements in the manufacturing so that we have a reduced price of goods. And currently, because of all the materials and very labor intensive, the cost of goods is much higher on a percentage basis for this therapy than for any other drugs. I'll add two other points. One way that some have advocated to reduce pricing is to go from so-called off-the-shelf products made from healthy donors, and those are in development by Allogene, Selectus, CRISPR Therapeutics, academic developers around the world, including ourselves. And I think that has potential, but recall that the immune system has evolved over several 100 million years to tell self from non-self. So it's currently possible to infuse those products and not have them rejected right away, but the length of time that those may persist so far is only about several weeks or perhaps a little bit longer. So we have to think in what setting might these types of off-the-shelf products be given? Well, my view currently is it's needed for some patients from whom we cannot obtain or we cannot generate sufficient cells, certainly in those situations. As improvements are made in the technology of knocking out HLA and inserting other molecules to make them invisible to NK cells, I think we'll see some improvements in durability. And then looking longer term at the horizon, the development of induced pluripotent stem cells, CAR-NK and CAR-T cells derived from iPSCs, I think has the potential to generate many, many doses that hopefully, potentially, would be less susceptible to rejection and reduce costs as well.
Thank you very much, Bruce. And if there are no questions anymore, no comments, I want to thank both of the speakers.
Marcelo, posso fazer comentário?
Claro, please, Sabrá.
Não posso deixar encerrar uma reunião brilhante como essa, não é? Agradecendo ao nosso presidente essa oportunidade dessa reunião e dizer ao Marcelo Morales e a você próprio que é uma honra muito grande ver a nossa participação nessa ciência com grandiosidade, não é? Podendo ver a nossa prima-dona brilhando como ninguém, como a nossa Patrícia Rocco, né? Muito obrigado pela oportunidade dessa reunião.
Eu é que agradeço, muito obrigado, colega Sabrá. Então, voltando a abrir a possibilidade de comentários e perguntas, e vendo que não há mais nenhum comentário, nenhuma pergunta, I wish to thank both speakers of the last session. It was a very exciting session. To thank everybody that followed the session, and the colleagues from the Academy and the visitors. And I pass the floor back to our President Rubens Belfort Jr. But again, I want to thank Marcelo Morales for organizing this fantastic session. Thank you very much, Marcelo. And please, Rubens.
Bom, todos os agradecimentos já foram feitos e claro que todos nós concordamos com o sucesso dessa reunião. Meus parabéns também. On behalf of the National Academy of Medicine Brazil, I want to especially thank our foreign speakers for being here and sharing their knowledge with us. It's been a great opportunity, and I want to tell you that we really appreciate your time and your presence among us. Thank you very much. Então, em nome da Academia Nacional de Medicina, nós encerramos esta reunião e desejo a todos uma ótima semana e que nos encontremos na próxima quinta-feira para a nossa grande reunião, como já foi anunciada pelo nosso secretário-geral, Ricardo Cruz. Boa noite, está encerrada a reunião. Boa noite.
Boa noite. Thank you.
Thank you.